Okay, everyone. Well, it's 12 o'clock, so let's go ahead and get rolling here. Um, so I am recording this, and I'll post that recording sometime later today. The way I'm going to run this, I want to go ahead and just point you to this poll everywhere poll. I think this is probably the easiest for me in terms of answering your questions. So this is active. It looks like someone's already answered or asked a question. That's great. So go ahead and pull out your devices and start putting your questions on here, and I'll work my way through as many as I can. As you put them on there, people can vote for them, and that will put those ones that are most commonly said, this is a question I have, towards the top. So, all right. So let me go ahead and just start at the top and then go from there. So it says this top one says, let me put this in full screen because I'm having a hard time reading it. So let's look at this top one. Go over the short answer on the practice exam. Okay, so let's do that first, and then hopefully questions will start to filter in here. I'm gonna go back to my Teams meeting. I'm gonna go ahead and stop my video, just because you'll be staring at the ceiling if I don't. So okay, there we go. So so go over the short answer question on the practice exam. So let's go ahead and go to the practice exam here. I've got it pulled up in anticipation that there will probably be some questions on this. Scroll down to the short answer question. So the short answer question says this: low blood glucose is detected by the pancreas that subsequently releases glucagon, a hormone that has widespread effects, including the activation of gluconeogenesis in the liver. Given this information, predict the effects gluconeogenesis activation on the following processes in the liver. Okay, so that's our question. Now, before I go on and answer this question, I forgot to mention this. You feel free to, by any means, go ahead and interrupt me, right? I mean, you can turn on your mic and interrupt me any way you want, especially if there's some like malfunction that you're experiencing. So, but if you have questions that you want me to, that go on with what we're talking about, feel free to interrupt, okay? Also, make sure you put something in the chat, just like, okay, I'm leaving now, or hi, or whatever you want to put in there, and that way I can look through the chat. Anyone that makes a comment in that, I'll give credit for being here, and that will go towards that extra credit for attending Q&A sessions, okay? All right, so let's look at this. So our context is this, essentially, and that is the gluconeogenesis has been activated, okay? And so just as a way to kind of diagram what needs to happen here, when we think about it this way, gluconeogenesis is going to do this. So it's going to take pyruvate, and I'm going to draw this in, a very non-detailed way, right? But it's going to take 2-pyruvate, and it's going to make glucose from those 2 pyruvates. Okay. Now, as we know, it'll need ATP equivalents to do that, a total of 6. Some of these are GTP actually, but we'll call them ATP equivalents. Right. So that's some of the energetic of gluconeogenesis. I'm getting some feedback here. If you have your mic, maybe make sure you're muted, and that way I don't quite get that feedback. So, okay, there we go. I think that was solved. Right now, the other thing then that we know has to happen here is that it has to have NADH, or a supply of NADH. Okay, and that would be for the GAP-DH reaction running in the reverse. Okay, so to continue this process, you need, of course, energetically, you need the ATP, but you're also going to need the NADH. Okay, and so when we think about this, the cell has to then supply that NADH in some way, and the way that it does so, and again, I'm going to draw this in somewhat abbreviated form, okay, but it will actually take pyruvate, convert it to malate, okay, actually, I'm sorry, I'm not going to take pyruvate and convert it to malate. Sorry. To convert it to oxaloacetate. I got ahead of myself. Oops, there we go. Convert it to oxaloacetate in the malate aspartate shuttle, okay, and part of gluconeogenesis with the pyruvate carboxylase reaction. And then, interestingly, that oxaloacetate will be converted to malate. Okay, and to do that, that's going to actually occur in the mitochondria, in the matrix of the mitochondria. Okay, and that's going to take NADH to do that. Okay, now that malate will actually be shipped out of the mitochondria into the cytosol, and it will be converted right back to oxaloacetate. Okay, so let me go ahead and kind of put a little box around this and say that this occurs in the mitochondria. Okay, and then that oxaloacetate will then make it out into the cytosol, and it will be converted. Oops, wait. Yep, this process right here, conversion back to oxaloacetate, that will occur in the cytosol. Okay, and the NAD8, the NAD plus, okay, that we see here will actually be used as the electron acceptor, and it will make the NADH. Okay, so the malate will take those electrons, give them the NAD plus, and make the NADH needed. Okay, and then this will go on and feed into, right, the oxaloacetate will feed into gluconeogenesis. Okay, so that's what's going on here, and the things that we see here in these two boxes, these are essentially the malate aspartate shuttle. Now, I didn't draw it all out, but that's what's happening, the malate aspartate shuttle. Okay, so there's obviously a lot of detail that I've left off, but that's a very important context to answer this question, because the question says this. The first thing is this: we've increased flux through gluconeogenesis, which means that we've increased our need for NADH, right? So then it says, well, the rate of malate production in the mitochondria, will it go up or down? Well, in the mitochondria, it's going to go up, okay, because the pyruvate's going to come into the mitochondria, it's going to be converted to oxaloacetate, and then that oxaloacetate is going to be converted to malate. Okay, so that's going to go up, right? That malate is going to be shipped out into the cytosol and converted back to oxaloacetate. Okay, right? So that's the first thing. Now, glucose export, excretion, or excretion out of the liver cell will be going up or down. This would be going up, right? So that's the whole purpose of increasing gluconeogenesis, so that glucose can be excreted into the bloodstream. So that's going to go up. And then finally, it says this. Predict the effects that a cytosolic malate dehydrogenase inhibitor would have on the rate of glucose production in the liver. Okay, would that go up or down? Explain why you think this is the case for part C. Right, well, here's the key to understand that question. This enzyme right here, let me get a good color here. This enzyme right here, that is malate dehydrogenase. Okay, the enzyme that carries out that process. And so what we're saying is this: predict the effects of the cytosolic malate dehydrogenase inhibitor would have on the rate of glucose production. Well, if we're going to inhibit this process, right, that will cause problems. And the reason that's going to cause problems is because we're not going to get then the needed NADH for the gluconeogenesis, right? The NADH supply will be shut down, right? And so then it says predict the effects of the cytosolic malate dehydrogenase inhibitor would have on the rate of glucose production in the liver. It would go down because you can't get the needed NADH. Not to mention the fact that you can't get the oxaloacetate either. And the oxaloacetate is going to be converted to PEP, and then the PEP is going to be used to make the glucose. Okay, so there would be two problems with this. You want to get the needed NADH, and you want to get the oxaloacetate. that would be needed to make PEP, or phosphoenolpyruvate, needed to make glucose. So, okay. All right. So that's the first question. Okay. Any questions on that before I walk away from that? Go ahead and.
I had a question on that. The process that you just drew out, is that reversed when we're talking about needing NAD plus for glycolysis, like where it's occurring in like the cytosol and mitochondria?
Yeah, great question. So the question is, okay, well, what if we had all these arrows going in the opposite direction? Would that be the reverse of that? And yes, it would be, right? So then in that case, let me go ahead and do this. I'll pull up a blank sheet here. What we'd have here in that case, if you're running glycolysis, right, in that case we have glucose being converted to 2-pyruvate. Okay, and of course in this process we know that, and we'll just draw this out as being 2 ADP or 2 ATP are going to be made. That would be the net. Okay, and then we know this, that 2 NADH are going to be made, and you'll need NAD plus. Okay, so then when you think about this from the standpoint of, okay, well, how does the cell then carry out production of NADH? Well, one of the things that it can do actually would be to take oxaloacetate and convert it to malate. So we can have this. Right, and that NADH would then be resupplying the NAD plus. Okay, and then I'll just draw ... Right, the rest of the process would go on the malate aspartate shuttle. So everything I drew for gluconeogenesis is the reverse of what would be happening for glycolysis. Yeah, so good question. Yeah. Other questions?
Yes, just to clarify one more time. So the cytosol and the matrix can change their action of malate dehydrogenase just based on if they need pyruvate or glucose?
That's right.
Okay.
Yeah. Yep, and the reason that is the case is because malate dehydrogenase is an enzyme that holds that reaction, oxaloacetate to malate or malate to oxaloacetate, at equilibrium. And so it's all based off of Le Chatelier's principle, right? So if oxaloacetate concentrations, or in this case, it would be NADH concentrations, because that's part of a reaction. If NADH concentrations are going up, then that will shift this reaction in this direction. Okay, or in the other case, if NAD plus concentrations are going up, then that can shift it in this direction. Okay, so it's all based off of Le Chatelier's principle.
Okay, thank you.
Yeah. Other questions from that little series? Okay. All right, so we've got that one completed, so we'll get rid of it. All right, so then let me look at this. How metabolic pathways are regulated and the effects each one has on kinetics. Okay, all right, so let's go ahead and look at that here. Both chapter 10 and 11, we talked about how metabolic pathways can be regulated, and there's really 5 ways that we talked about in terms of regulating a metabolic pathway. Okay, the first one was to change genetic expression. Okay, meaning that you could increase or you could decrease the amount of the enzyme that catalyzes the rate limiting step of the reaction. Okay, and if you're increasing that, the rate limiting step, not of the reaction, but of the metabolic pathway, I'm sorry. But if you're increasing that enzyme in concentration, you're going to increase the rate or the flux through that pathway. Okay, if you decrease that enzyme concentration, then obviously you're going to inhibit or decrease flux through that pathway. Okay, so that would be one. We didn't talk too much about mechanism to do that, but we did mention that. Okay, the second one that we talked about is that enzymes can be bound to what would be considered regulatory proteins. So regulatory proteins can bind to an enzyme and either inhibit or activate them. And so then when you have, right, a need to inhibit some particular pathway, a regulatory protein can bind to the enzyme that catalyzes the rate limiting step of that pathway and can slow it down, okay, or vice versa. Right. So that would be number two. Number three would be to sequester the enzyme in question, right? So if there's some enzyme that is catalyzing the rate limiting step of a pathway, then you can actually take that enzyme away from the compartment in the cell that that enzyme or that pathway runs, and that would then inhibit the pathway. Okay, hexokinase is actually an example of that. We didn't really talk too much about that in glycolysis, but hexokinase catalyzes the first step of glycolysis. And one of the means of regulating glycolysis is to remove hexokinase from the cytosol. It's called sequestration. Okay, again, we didn't talk too much about it, but that's another way. Okay, so right now we've got change expression of the protein, increase it or decrease it. We've got bind to regulatory proteins. We've got sequestration. Okay, so there's three. And then the last two are probably the most common ways of doing this, and that is through either allosteric means or through covalent modification. Okay, and allosteric means would be essentially what we talked about with hemoglobin, but now we're talking about enzymes. And so with allostery, we can have either a positive or a negative modulator of the enzyme. A positive modulator will, of course, increase the activity of the enzyme. A negative modulator will, of course, decrease the activity of that enzyme. Okay, and if you're, of course, decreasing the activity of that particular enzyme, then you'll decrease the rate through a particular pathway. Okay, and then finally, covalent modification can be either inhibition or activation as well. Covalent modification is just simply attach some small molecule group covalently to the enzyme, okay, and that can either have an inhibitory or an activating effect, and is entirely context dependent. Okay, so those are the five main ways that we talked about in terms of how to regulate a particular pathway. Okay, and we talked about that in chapter 10. In chapter 11, we then gave some specific examples using glycolysis and gluconeogenesis. Okay, so for instance, ATP can allosterically inactivate PFK1 or allosterically inhibit PFK1. PFK1 catalyzes the rate limiting step of glycolysis, and if you then have increasing ATP, in fact, let me go ahead and draw this out so we can give you this example here. So let's do this, because this will give us a good understanding of this. Okay, so you have glucose that's converted to glucose 6-phosphate. That's catalyzed by hexokinase. Then it's at equilibrium with fructose 6-phosphate. And then fructose 6-phosphate can be converted to fructose 1,6-bisphosphate. And then there's a bunch of steps. Okay, and so I'll just do it this way. There's a bunch of steps that are held in equilibrium. Okay, and that will eventually lead to PEP. Okay, and that'll be 2-PEP, and then this will be converted to pyruvate irreversibly. Okay, and that's glycolysis. And you all say, oh, why didn't you just present it that way the first time? So I know, I get it. All right, but in any case, of course, we know that this whole pathway, one of the major products of the pathway is going to be this, right? And I'll just have it coming out, is going to be ATP, okay, fire molecule. Right? Well, it turns out that ATP, right, so this enzyme here, the enzyme that catalyzes this step right here is PFK1. Okay, well, ATP can actually feed back and inhibit that. Okay, that would be through allosteric regulation, and that will then slow down the consumption of glucose through glycolysis, because there's a feedback mechanism saying, oh, there's a lot of ATP. So if this is going up in the cell or ATP is not being used, then there's no good reason to continue to consume glucose through glycolysis, and therefore PFK1 could be inhibited. Okay, that depends on the cell type and what we're talking about, but if you're in the muscle cell, that makes a lot of sense. Okay, and liver cell is a little bit different, but anyway, okay. So let me come back to that question. It seemed like there was a couple points. How metabolic pathways are regulated. So we got those 5 ways, and the effects each one has on kinetics. Okay, maybe a little bit more specifically with it has on kinetics, right? So that really what we're thinking about there is rate. But let me go ahead and maybe try and illustrate a little bit more about what we just talked about. Okay, so if we were to think about this from a kinetic standpoint, we can look at PFK1 actually. Okay, so if we were to do PFK1 activity or rate on our y-axis, and then we were to plot that against our substrate concentration, and actually let's be specific here since we're talking about PFK1. Our substrate is fructose 6-phosphate. Okay, and so what we'd have here in regards to PFK1 activity, you would see something along these lines, or could see something along these lines. Okay, 2 different models of PFK1 activity. That one, and then something more akin to or likened to this. Okay, both of them approaching the same Vmax. That's a Vmax. Okay, but these are obviously 2 very different curves, right? And that blue curve here, this curve here, would be PFK1 activity at high ATP concentration. Okay, and then this other curve could be at low ATP concentration. Okay, and then if you were to consider this just from a, right, so this is our kinetic standpoint, really what's changing here will be, it's K half, K 0.5, it's not really the Km, but it's very, very similar, if not identical concept. Okay, these are not Michaelis-Menten enzymes because they don't have hyperbolic behavior or saturation profiles, they have sigmoidal saturation profiles. But essentially what's changing here is the affinity or the specificity of PFK1 for its substrate, fructose 1,6-bisphosphate. Okay, so if we were to look at it like this, let's say that we have, right, some concentration of fructose 6-phosphate in the cell, a steady state concentration, we could say this. Draw a straight line up here and say that that is fructose 6-phosphate concentration in the cell. Well, you can see what's happening here. When we are at low ATP concentration, oh wait, shoot, big red check minus. My goodness. I'm sorry. I hope someone was about ready to interrupt me, or you're like thinking to yourself, what's going on? This should be at low ATP concentration. Sorry. This is at high ATP concentration. Big mistake. Sorry about that. I caught it. Some of you are wondering. So this is at high. There we go. High ATP concentrations inhibits. Okay, so we caught it. Okay, but now we can go to this. Let's look at this. Okay, so at high ATP concentration, what you'd have is a really low PFK1 activity. Okay, at the fructose 6-phosphate concentrations in the cell. And then up here, right, we have then when ATP concentrations all of a sudden go down because let's say you took off on a run or something like that in your muscle cell, then your PFK1 activity is going to go way up. Okay, you're going to lose that inhibition. And therefore that will change the rate of the entire pathway, and there will be more ATP production because quite honestly your cell needs it at that point in time. So, okay, right. And so this would be an example where K half is being changed. You can have examples where K cat is being changed, meaning the Vmax would change. That will be another way of doing allosteric inhibition. So, okay. Right. Questions on that? I know I screwed up in the middle there. I got a red check minus. That was a big one. Any questions on that, though? No? Okay. Let me go ahead and get rid of this question then. And we'll do the next one. Is the LDH cycle included in gluconeogenesis? No, it's not. Well, yes, it is actually. So now that I'm thinking about it, so yeah, let me go ahead and draw this out. Okay, so when we come back to this, okay, I'll draw a very similar picture here that I had before. That can make glucose. And again, all of you kind of groan and say, why don't we just do it that way in the first place? Because it's not like nearly as cool, right? So 6 ATP, again, equivalents that have to be used as the energetic needs. And then the NADH, of course, 2-NADH, still needed to do this process. Right, so in my last depiction, okay, I can go there. Let's go there, right? So I had all of this stuff going on, and there's this malate aspartate shuttle and the whole 9 yards. Well, it turns out that you can actually have, you can actually do this. The cell will absorb, the liver cell, because the liver cell is what does gluconeogenesis, can actually absorb lactate from the bloodstream, okay? And use LDH, lactate dehydrogenase, and convert that to pyruvate. Well, to do that, you need NADH that will resupply NAD+. Okay? And you think to yourself, man, that's a lot less complicated than that last diagram. Why doesn't the cell just do it that way all the time? Well, it does both. So, and then you say, why? And I say, because. And that's a good answer for that. Okay, so there are multiple reasons, I'm sure, but because is an okay answer for that. Okay, so yes, the answer is the lactate cycle will be used in this. So, yep. And by the way, just so we know, right, lactate, this can come from, just to expand your horizons, this can come from the blood, right? So lactate is in supply in the blood because a lot of our cells will metabolize glucose to pyruvate and then convert it to lactate, and those cells will, generally speaking, excrete it. Okay, and lactate can be absorbed by several different tissues, actually, and used for different reasons and different purposes. Cardiac muscle, for instance, will use lactate from the bloodstream as an energy source. It'll convert it back to pyruvate and use it in cellular respiration. In fact, it's one of the preferred metabolites of energy metabolism in the cardiac muscle. Okay, so that can certainly come from the blood. Another source of this, though, can come from amino acids. Okay, actually, but yeah, amino acids typically will actually be converted to pyruvate. Okay. But amino acids can also be converted to lactate as well. So, okay. All right, let's go here. Okay, so answer that one now. Okay, can we go over the aminotransferase portion of the malate aspartate shuttle? Sure, let's do it. So, malate aspartate shuttle, so much fun. I have to do that. Let's see here. Okay. All right, so what do we have here? Let me put my bearings on this, okay? So I'll draw it in a way that you've sort of seen it depicted to you, okay, in the book and also in the Echo 360 recordings, okay? But you have oxaloacetate, okay, that's converted to malate. Okay, that will then produce, you have to have NADH to do that. Okay, so that's going to produce NAD+, which is really important. okay? Right, and then you're going to have, of course, we have to have a mitochondrial membrane, okay? And we'll have to have a transporter in that membrane, okay? And then we'll have another transporter down here, okay? And the malate will then get shuttled into the mitochondria. So this would be the matrix in here, okay? And malate will then get into here, okay? And then the oxaloacetate will be regenerated, okay? And that's going to then require NAD+, and that's going to make NADH, okay? Now, incidentally, because I've had this question in emails and stuff, what's the purpose of this NADH here? Well, that will go on to the electron transport chain, okay? And that's going to make ATP. Okay, so that's an important purpose of the malate aspartate shuttle, okay, is to get that NADH. Notice that the NADH here gave its electrons to malate, right? And then malate is moved, and those electrons are given right back to NAD+, and then the NADH can go on and make ATP through the electron transport chain, okay? Right now, this is the part that was being asked about, the aminotransferases. Okay, so what's going to happen next? Well, oxaloacetate can't get out of here. Okay, it can't get out of the matrix of the mitochondria. There's no transporter. So what the matrix does is it will convert it to aspartate. Okay, and aspartate, you think to yourself, man, that sounds like an amino acid. That's because it is an amino acid. Okay, you are right. It is an amino acid, and oxaloacetate can be converted to aspartate, but it needs an amino group. Okay, so to do that, okay, an amino group meaning the alpha amino group. Okay, and so what's going to happen here is this. I have to choose my colors carefully, but there will be another amino acid, glutamate. Okay, and glutamate can give up its amino group and become something called alpha-ketoglutarate. Okay, alpha-ketoglutarate, we'll find out in chapter 12, is a part of the citric acid cycle. Okay, as is oxaloacetate. Right, so what is essentially happening here is that the alpha amino group on glutamate here is being transferred to oxaloacetate to make aspartate. And then aspartate will be shipped out, okay, to here, and it will be converted right back to oxaloacetate. Okay, now, how did it get back to oxaloacetate? Well, it had to give up its amino group, its alpha amino group, right? It gained, oxaloacetate became aspartate beginning in an amino group, okay, from glutamate. And so for aspartate to go back to oxaloacetate, it has to give up its amino group, right? And it's going to give up its amino group to alpha-ketoglutarate. So alpha-ketoglutarate will be out here, and it is going to grab that amino group and become glutamate. Okay, now all we have to do is just complete our shuttle then here. Every time aspartate moves out, glutamate moves in, okay, and every time malate moves in, alpha-ketoglutarate moves out. Right, so that would be, if the question was, you know, can you go over the aminotransferase reactions? That's what this is. Okay, the aminotransferase reaction. The name of the enzyme actually gives away what it's doing. It's transferring transferase an amino group, aminotransferase. Okay, and it's transferring the amino group from glutamate to oxaloacetate to make aspartate, or it's transferring it from aspartate to alpha-ketoglutarate to make glutamate. And you said, what? Aspartate to alpha-ketoglutarate to make glutamate? Man, only in biochemistry would we get a sentence like that. And that's true. Only in biochemistry would you get a sentence like that, okay? Right. Hopefully that makes sense.
Sorry, just a really quick clarification. Can you just repeat what you said at the last part? Like the malate like goes in or something, and then the alpha keto KGA goes back in. Like, sorry.
Yep. Yeah, no, it's okay. I completely understand. I'm just drawing these arrows all over the place and all that kind of stuff.
Just one more time.
Yeah, one more time. So what I said is this, right? So I think that what you were referring to is I didn't quite have these arrows here yet. Okay.
Yeah, that.
Yep. And so what I said is this, okay? These transporters, let me just put a star by each of these boxes here, are antiporters, okay, to draw on our chapter 9 material. Antiporters move things in opposite direction, right? So it turns out that every time an aspartate goes out of the cell through the bottom transporter, a glutamate will come in. And I said through this out of the cell, I meant out of the matrix. Okay, a glutamate will come in at the same time, right? So that your every aspartate that's generated and moved out, you move a glutamate back in. Okay, and the purpose of that is because to make that aspartate, we have to have a glutamate, right? So if we remove an aspartate, we better replace it with another glutamate. Okay, now the other thing then I said was this, coming to the top antiporter, okay, that will move a malate in, and every time we move a malate in, an alpha ketoglutarate or alpha KGA goes out, okay, for similar reasons, right? So this is just to make sure that the things on each side of the membrane are balanced, okay, and those antiporters do that.
Okay, that makes more sense. Thank you.
Yeah, no problem. Other questions? Great question.
Just to be clear, so what's it called? OAA, is it using glutamate in order to convert it to aspartate?
It is using yes, to a certain extent. It's using the amino group on glutamate to become aspartate. Right. So
gotcha.
Let me do it this way. Let me kind of show you this to help you out. So when we think about aspartate, right, and this is where, quite honestly, it can be helpful to know structures, because the chemistry kind of bears itself out when you know these structures. But this is aspartate, okay? And you all said, yes, of course I remember that. I memorized that long ago, okay? So, but yeah, that's aspartate. Well, when we look at oxoacetate, let me draw it in the same way, okay? So oxoacetate is going to be this. Okay, so that's oxoacetate. So you'll notice here, hopefully what you notice here is the difference is this, right? So, oops, I'm sorry. That would have been a red, should be another red check minus. This should be, there we go. Okay, right, but the difference there is the amino group, okay? So when we come up to here, this oxoacetate has to become this. Well, it has to gain this amino group. And it's going to gain that actually from glutamate. So if you remember, glutamate structure is this. Glutamate and aspartate look really, really similar. Here's the amino group on glutamate, okay? And glutamate, the only real difference between glutamate and aspartate is a single carbon on the side chain, okay? And so for this, be clear here, oxoacetate, to become this, it's going to take this from glutamate. So this is glutamate here, okay? And so that's what's happening. Now, then glutamate becomes alpha ketoglutarate. Well, what's alpha ketoglutarate look like? Well, Let me show you. So alpha ketoglutarate is this molecule. It is essentially glutamate without an amino group. So that's alpha ketoglutarate. Okay, and again, notice that the only difference between glutamate and alpha ketoglutarate is an amino group. Right. So those are our molecules that we're looking at, and of course, I wouldn't expect you to memorize all that and have that for the exam, but that can be helpful when you start to look at this, right? What's going on here? Okay. Other questions?
Could you go over how the malate aspartate shuttle works in gluconeogenesis?
Yeah, so I kind of already did that, but let me. So that is essentially this. Okay. So trying to answer this question, let me go ahead and bring it back into this context, though. Okay, and I'll use it here. Essentially, what we have here, I'm going to have to change some arrows on this. So let me get rid of some of these arrows here. There we go. Okay, there we go. Get rid of all that stuff. Okay, so everything that I did, actually I got to get rid of this too. So everything I did with before in the last one, I was just showing what was going to happen when glycolysis was occurring. Okay, we were assuming glycolysis was going on. When we start to think about gluconeogenesis, these arrows just start to go in the opposite direction. Okay, now it can get a little bit funky, and I don't want to get it too funky before the exam for you. Okay, this is meant to clarify things and not mystify things. But let me just go ahead and show you this with gluconeogenesis. Okay, and I'll use pink molecules or pink to represent what's happening with gluconeogenesis. Okay, so pyruvate, we know this. Okay, and the reason it can get funky is because of stoichiometry. Pyruvate we know that the very first step of gluconeogenesis is the production of oxoacetate from pyruvate, right? So that's the very first thing that will happen. The enzyme is pyruvate carboxylase, and this would be the pyruvate kinase bypass reaction, right?
So is there just like a store of pyruvate in the matrix then?
No, there's not. So you'd have to get that pyruvate from somewhere, right? So you would get that pyruvate from typically amino acids or lactate. Okay, so that was what we're kind of doing up here. Okay,
thanks.
So you get that pyruvate from lactate or amino acids. Okay, in this case, right, when we get it from lactate, you typically don't do the malate aspartate shuttle. When you get it from amino acids, you do this kind of thing. So we could say amino acids are converted to pyruvate. Okay, in fact, 18 of the 20 amino acids can be used to make glucose. Okay, not all of them will go through the route of pyruvate, but many of them will. Okay, right. So anyway, when you do this, right, so pyruvate is converted to oxoacetate. That's the first step of gluconeogenesis, right? Now, that feeds, as you can see here, it feeds right into the malate aspartate shuttle. Oxoacetate can be converted to malate. NADH can be converted to NAD+, and then this can be shipped out this direction, right? And this is why I erased all my arrows. And then malate can be converted back to oxoacetate, okay? And to do that, that's going to make NADH. Okay, now understand that NADH is what's needed, right, for gluconeogenesis. Now, this then will go on and be converted to PEP. Okay, and that's the second step of the pyruvate kinase bypass reaction, right? So that was the enzyme it's fun to say and spell, the phosphoenolpyruvate carboxykinase. I like Pepsi K better, right? But that's Pepsi K, okay? And then this will be converted, right, through multiple steps to glucose. Okay. Right, so the malate aspartate shuttle is part of this. Now, this is where, and I don't, like I said, I don't want to mystify things for you. I want to keep them clear. But stoichiometry can get kind of tricky with this, okay, because the malate aspartate shuttle will continue to go on in the way it does, right? So oxaloacetate can be converted to aspartate. Aspartate can be shipped across, and then that can be converted back to oxaloacetate. And then to do that, we would actually do it this way. Alpha-ketoglutarate would pick up the amino group and become glutamate, okay? And the glutamate would give up. Out here, it would give up its amino group and become alpha-ketoglutarate, right? So everything would be kind of running in the reverse direction from what we talked about, okay? And you would have to really watch out for your stoichiometry with this. You don't need to have the stoichiometry exactly right to understand this connection, though, okay? But what you see here is this in the pink, and this is why I said I use pink. In the pink, right, this is all part of gluconeogenesis, and you kind of see in here that, oh, malate aspartate shuttle is kind of this in-between step of getting pyruvate out of the matrix into the mitochondria to make Pepsi K and also the NADH. I'm not Pepsi K, but PEP and the NADH is needed. So, okay. Hopefully that helps out.
Excuse me, Dr. Shoulders. So you said NADH is what is needed for gluconeogenesis. Can you remind me again what NAD plus is needed for over in the matrix?
Yeah, so NAD plus is needed for like glycolysis.
Okay, gotcha.
Yeah, it's an electron acceptor, right? So,
okay.
And so when we think about gluconeogenesis and we think about glycolysis, so this is all hinges on this reaction. Let me go here. I'm running out of space, but that's okay. I'll erase this stuff on here. Don't need it anymore. So when we think about NADH and NAD plus, since we spent a lot of time talking about this over this review, right? We've got this reaction that takes glyceraldehyde 3-phosphate and will make, in one direction, 1,3-bisphosphoglycerate. Okay, and to do that, you have to have an electron acceptor. Let's see here, NAD plus. Okay, that'll make the NADH. Okay, and that's part of glycolysis.
Okay.
Okay. And then part of gluconeogenesis, the opposite of this, where all I have to do now is just say, okay, well, I have to have an electron donor. NADH is electron donor. And that's going to regenerate NAD plus. Okay. This is part of gluconeogenesis, right? And the enzyme that does this is the same enzyme in both of them. It's reversible. Okay, but the enzyme that does this is GAPDH, or glyceraldehyde 3-phosphate dehydrogenase. Okay, and so all of the discussion on the malate aspartate shuttle and even coming up to here, the lactate and all this stuff, and on gluconeogenesis and glycolysis and all that jazz that we've been talking about centers on this reaction here. Where you're either needing NAD plus for glycolysis or you're needing NADH for gluconeogenesis. Okay?
Gotcha. And then I was also wondering, because on the quiz last night, we were talking about pyruvate, and I can't remember. Can we review real quick the energy investment and energy generation phases? I think one was 1,3-BPG and the other one was pyruvate.
Yeah. So just in terms of review of those, let's see here. Let's see if I can't find a blank space. I don't want to erase that. I'll use this one. get rid of all this junk. Okay, great. All right, so when we think about energy investment, the energy investment phase essentially is glucose, okay, is converted to, and let me just do it big. I'll just draw it as kind of one big step. Glucose is split into G3P. 2 moles of G3P, glyceraldehyde 3-phosphate, okay? And that's really the first 5 steps of the pathway, okay, and the first 5 enzymes. And we call it energy investment, the reason being because you have to use 2 ATP to do it.
Okay.
Right, so, in fact, really good D, there we go. You see these P's here, the phosphates here, those phosphates come from ATP, right? So we talk about glyceraldehyde 3-phosphate. Well, those phosphates came from the ATP, okay? Now, these then, okay, in the energy generation phase, we'll boil this down to one big arrow, okay? These are gonna make 2-pyruvate. And that's gonna make a total of 4 ATP.
Okay.
From ADP. Oops. There we go. So energy generation, meaning that we have now generated the ATP, right? At the very beginning of this phase, okay, is that last reaction that we were just talking about, and that's where the NAD plus is converted to NADH. Okay, right. So that's the 2 phases. That I think is as good a review as you. I mean, as just kind of tertiary, but that you would need. So, okay. Right.
So for number 56 through 60 on the practice exam, it's just relating to what you're talking about here.
Yeah, let's just go for it.
I'm sorry, go ahead.
No, I just said go for it. Yeah, ask your question. What's up?
Okay, so it talks about the mole ratios of the NADH and ATP. So in the question it says there's a net formation, but then it says this does not take into account what happened in the prep phase. So, I mean, I know the answer is 4 mole of ATP. So I guess I got a little confused because it says net formation, but then we're not talking about the 2 ATP that were used before.
Well, yeah, let's look. So it says the conversion, and so this is using terminology, energy investment and energy generation. And I see what you're saying, I think. So it says the conversion of 2 moles of glyceraldehyde 3-phosphate to 2 moles of pyruvate by the payoff phase in glycolytic pathway results in the net formation, right? So understand what the question is saying. It starts at G3P, okay? And it ends at 2 moles of pyruvate. Okay, and so when it says net, maybe it's not a great question, but it is talking about. Let's go back here. It's talking about essentially this. Right, so that portion. So 4 ATP to an ADH would be the net of it. Okay, and so it's kind of a. Yeah, I mean, for a practice exam, it is all right, but I would hope to do a better job on a question like this, because I think what's kind of tripping you up is this idea of net, and you're thinking to yourself, okay, well, that means I need to subtract what happened in the payoff phase, and that's why there's, in the prep phase, or energy investment phase, and it says this does not take into account what happened in the prep phase, so you're only left with this one answer. So does that make sense to you? I don't know if that answered your question.
It does clarify it. Thank you.
Yeah, not a great written question. I would agree with you on that. So, but hopefully you can understand conceptually what the answer is. So
Yes, thank you.
So just to clarify, the prep phase is the energy investment phase and the payoff phase is the energy generation phase.
yeah, and I will make sure that in the exam and I have made sure in this exam that I only use energy investment and generation because that's the terminology that we've used most often in the class. When I introduced it, I did say, oh, sometimes this is called prep and payoff phase, but the way you just said it is right. Prep, sometimes the energy investment phase. Let's write this out so we don't rely on just words. Verbal or spoken words, but let's do this. This would be the payoff phase that I have written here, which is what you were saying. Okay. And then this here would be the prep. And so it really depends on what textbook you're looking at. Our textbook uses energy investment and energy generation, whereas many other textbooks will use prep and payoff, right? In fact, you may have taken another class where you had the prep and payoff, or may take another class where it's prep and payoff. So, okay. All right. Other questions?
Yeah, Dr. Shores, back to chapter 9 regarding the beta barrels. I'm just trying to remember, is the inside of the beta barrel, is it allowing, I just can't remember, is it there to move proteins through the membrane due to it being hydrophilic or due to it being charged, or is it like kind of a combination of both?
Combination.
Okay.
So, yeah, the beta barrels, quite honestly, are probably the least selective of all the membrane transporters that we would talk about.
Okay.
They're more like channels, but they're really big channels, and they're not very selective at all. In fact, many beta barrels are essentially holes in a membrane that just have some molecular weight cutoff that just allow polar things to move through them. Very rarely would you find a beta barrel found in a plasma membrane, like the membrane that surrounds the exterior of the cell, because typically that would disrupt the entire set of gradients that the cell is trying to set up with all the other transporters. So they're used sparingly in that. You do see them in the mitochondria, though. The outer membrane of the mitochondria has a really big, what's called porin. That's essentially a beta barrel. And it's just this big hole. But it allows, polar things through. Yeah.
Gotcha. So would we only see beta barrels in the outer membrane of the mitochondria and not in the inner membrane and in between the inner membrane and the matrix?
I mean, I don't have an exhaustive knowledge of all the transporters in the inner membrane or in the plasma membrane, but my intuition would say yes. Typically, beta barrels are not very selective, and the inner membrane of the mitochondria is ridiculously selective. So it's, if you put a beta barrel in that, my assumption would be that you'd have no way to maintain an ion gradient that the cell works very hard to produce, to make ATP. We'll talk about in chapter 13. So that would be my intuition, yeah.
Gotcha. And then just real quick, so what's the difference between PEP and PEP-CK?
Well, PEP is phosphoenol pyruvate. It's a small molecule. And then PEP-CK is phosphoenol pyruvate carboxykinase. So PEP-CK is the enzyme that uses the small molecule PEP to make that uses oxaloacetate to make PEP. So I said PEP was its substrate. It's actually oxaloacetate that's its substrate to make PEP. Yeah.
So sorry, I'm just trying to repeat what you said. So PEP-CK uses OAA to make PEP?
Yep.
Okay.
That's right. Yep. Okay. All right. Well, everyone, it's 1:00 and I have another class that I need to get to. So I'm going to have to stop. Please make sure that before you leave, put something in the chat, and that way you can get credit for being here. I'll go through it and give you the credit, your Q&A credit. I did record this, so if you had questions that oh man, I need to go back and listen to that, by all means, go do that. Okay. And good luck on your exam tomorrow. Okay. I'm going to be not around my computer a lot tomorrow. Lots of things going on tomorrow. So hopefully you don't have a lot of issues. I don't expect there's a lot of issues, but I won't be able to get to my email very often tomorrow. So I'll check it certainly, but I'll kind of sparingly. So hopefully it goes off without a hitch. If you do have a hitch, let me just say this: an electronic hitch, right? Something that's outside of your control. Send me an email by all means. And if I don't get to it, don't stress out, right? Don't think, oh my gosh, I'm going to fail this exam because, I had this thing. I can give you extensions if need be. I haven't had any problems thus far, so I'm not too worried about it, but just in case, okay? All right, take care, everyone, and have a great spring break. I'll see you after the break, okay?